According to the Center for Disease Control and Prevention, over 2 billion people around the world lack access to safe drinking water, and this number is predicted to skyrocket to 5 billion in 2050. At the heart of this issue is Jakarta's Ciliwung River, one of the world's most polluted rivers. Due to the dumping of industrial waste, this river has become an ideal place for pathogens to proliferate, threatening the lives of the communities surrounding it. Our group decided to tackle SDG 3 and 6 to provide clean drinking water for the low-income people that depend on it for drinking, showering, washing, and many more daily life activities.
Our team, BlueManta, wanted to solve this issue by creating a low-cost portable water filter that would enable people to have access to clean water wherever, whenever. Our first filter layer was inspired by the filter-feeding lobes of a manta ray, which uses a spoiler orientation to effectively filter out macromolecules and sediment without clogging like conventional water filtration systems. This is done through a process commonly known as cross-flow filtration.
However, we still needed to create another layer to filter out the pathogens and microplastics from the water. First, our group looked towards the aquaporin channels in our cell membrane. But after meeting with the World Bank's water and sanitation specialist, civil engineer George Butler, we realized this idea was not feasible because we had to replicate the amino acids and protective layer needed for the aquaporins to function properly.
After conducting further research, we looked to mimicking plant xylem vessels to filter out the pathogens. The interconnected conduits with nanosized pores in xylem are structured to trap vapor bubbles. Our group uses property to filter out at least 99.9% of pathogens, making it the most effective and feasible way to remove the pathogens present in water. However, in order for the system to work efficiently and filter water at a fast rate, we needed to increase the hydrostatic pressure of the water entering the filter. To achieve this, we were inspired by the shape of the human kidney afferent and efferent arterioles, which have increased pressure due to the decreasing lumen diameter. To ensure the water filtered is as clean as possible, we also decided to make the container where water is collected transparent. This allows us to harness the UV rays from the sun, which can deactivate harmful pathogen activity.
Mr. Butler also inspired us to make the body of the bottle out of vitrified clay, which is resistant to abrasion, environmentally friendly, and strong. We also chose to make the collecting cup and lobes out of rPET plastic, which is also sustainable, durable, and flexible.